So welcome everybody. So today in the series of conference that we organize for the Industrial Partner Club of the GDR Imabio. So today is Zeiss company with which we organize this session, and Cyril Favard will moderate this session. So Cyril is a co-team leader at IRIM in Montpellier, and is also a member of the GDR Imabio steering committee. So Cyril.
Yeah, thank you. Thanks, Virginie. So thanks to the Zeiss company and Michael Gay, and also to Lorenzo Cipioni to have accepted to do this webinar today. This will be, as you know, splitted into two parts. First, Michael will talk about the last developments in Zeiss company about FCS and related techniques. And then we will have a scientific webinar by Lorenzo. So, Michael, I'll let you speak.
Yeah, I will share my screen.
Yes.
It should be okay now.
It is.
It is. Okay, good afternoon, everyone. I'm going to do a short presentation about a new functionality proposed by Eriscan detectors. As indicated by title, Eriscan is now able to give you some special information, and I'm going to explain it. LSM is perfect to give great performance in respect of speed, resolution, and sensitivity, and it provides one of its kind spectral flexibility. So instead of the one node triangle of dome, we have here a pyramid of opportunities. Turning now the fifth dimension of the imaging pyramidal, another way of looking at your sample is considering dynamic processes such as diffusion. One way of monitoring dynamic processes is using FRAP for fluorescence recovery after photobleaching, where fluorescence is bleached in a specific area of the cells, and the dynamics are extrapolated by how the fluorescently labeled molecules are repopulating this area. This technique is powerful and can address many questions, but come with compromise, as bleaching is harmful to the cells and repeat measurements are not possible. Using FRAP is always be an average of a large population of proteins, and additionally, also the fluorescence is lost in the region of bleaching, the actual protein is still there, and therefore the potential interaction and adherence cannot be assessed. Another way to address the study of molecular dynamics is use FCS for fluorescence corrective spectroscopy, a technique that is assessing molecular dynamics by monitoring the movement of molecules going in and out of the observation volume. This movement is recorded by the contrast, and the latter is used to mathematically extract information of different parameters of interest, such as diffusion, concentration, and number of molecules. While a precise and very sensitive method, traditionally, it is limited to extremely low concentration expression level or molecular concentration that can be well below the experimental expression level in live research sample. But FCS holds so many possibilities, as you can see here from this published overview. It is a great technique to get into molecular behavior and extract a multitude of data from your samples. So with FCS being part of the Zeiss portfolio for so many years and being used by the scientific community, it is no wonder that new ideas arise when new technology become available. So the new idea to use the spatial information of the Eriscan detector was published in 2018 by Lorenzo Cipioni, that we do the second part of this presentation. The inspiration led to the idea not only to use the Eriscan detector for several molecular dynamics options by adding spatial information of the measurement, but to rethink FCS. Accessibility was in focus, and when developing the dynamic profiler, we had the vision that it will work on all samples to have an easy to use tool. The third challenge was to make it available for all samples that you would normally use FRAP for. And we could show that now FCS measurements are possible on bright sample as well. We are rendering it possible by using Eriscan detector in normal analog mode, capitalizing on a great dynamic range, and then transfers the signals into equivalent photo count. For best signal to noise ratio, we decide to use only 19 inner elements of the detector, even though we collect all 32 elements in each measurement. Here I have the according dynamic profiler measurement, now delivering molecular concentration. Oh, sorry. Sorry for that. Yeah, I said that we are rendering possible by using. Oh, it was a slide after. I'm sorry for the recording. So here are the according dynamic profiler measurement, now delivering molecular concentration for all 4 cells, which have very different expression level. Contrast for the spot are recording during the measurement, and correlation curve are generated. Correlation curve are fit, and concentration can be retrieved as seen at the correlation table. In these examples, the difference of diffusion time of tetrameric GFP in a nucleoplasm and in a nuclei was investigated. Fibrillary marks the nucleoli in magenta and helps to position the spot. Normalization of the amplitude omits the difference in molecule number and thereby allow better comparison in diffusion time. The slowdown of GFP within the denser nucleoli can be read from the table and from the shift of the correlation curve. The diffusion time of tetrameric GFP is much higher on average in a nucleoli compared to the nucleoplasm, indicating that the denser structure of the nucleus seems to slow down the free diffusion of the GFP. Dynamic profiler also allow you to get information about flow measurement. The flow analysis measures the speed and direction of fluorescence molecule, all within a single excitation volume. It is perfect for a measurement of active movement of solution, such as biological processes in a bloodstream or in microfluidic systems, such as organ on chip. To analyze flow, not all detector elements are read out and correlate, but pair of 2 detector elements are used and then correlated with each other. This is called cross-correlation. Using this method, we can determine if molecule seen by one detector element will pass a bit later at another element and determine the direction of the flow. Here we measure the flow speed and direction of blood flow in zebrafish embryo. An overview scan of the blood flow on the blood vessel system of the larvae were generated at lower magnification. 3 spots were positioned within the vessel with the help of the reference image. The correlation table includes all the relevant parameters, including number of molecules, flow direction, and speed. Dynamic profiler also allow you to get spatial information of your molecule dynamics, such as asymmetric diffusion between different cell compartment, barrier, and at the interface of liquid-liquid phase separation leading to cellular condensate. To determine difference in molecular behavior to center detector element is cross-correlated with the detector of the third ring of the L scan. This is often called pair correlation. So along each axis of cross-correlation curve, along each axis, a cross-correlation curve is generated and then can be compared in regard to molecular number and diffusion time. The color code helps to visualize relative height and length of the correlation curve to make interpretation easier. Really sorry for that. I don't know what's happened. Yeah, to make interpretation easier, the hand map is representing exactly the space in the sample that is measured with the aeroscan detector. This way, heterogeneous molecular behavior in a single PSF without any delay can be recorded. These examples show HeLa cells that form GFP labeled stress granule upon each shock. The spatial information that we gain with the dynamic profiler is ideal to investigate cellular condensates that form by liquid-liquid phase separation. Condensates lack surrounding membrane, and measurement to uncover dynamics at the border during this information are therefore of special interest. The reference image helps to position the spot and to orient within the sample. Clear asymmetries matching the orientation of the granule in the reference image can be found at both border, while no clear asymmetric behavior was found within the granule. Spatial information collected by dynamic profiler allow to characterize heterogeneous diffusion behavior ideal to investigate the cellular condensate. The great thing about dynamic profiler is that all three analyses are included in the same measurement. You don't have to decide upfront what you want to do. And now, Aeroscan is the only detector that has so many different applications, delivering superior super resolution, speed through multiplex polarization, and no molecular dynamics measurements. So just to finish, if you want to learn more about dynamic profiler, you can scan the QR code to read the web page and the white paper.
Thank you. Thank you, Michael. Thank you. Are there any questions directly for Michael before we move on, or alternatively, I propose that you ask all the questions at the end of the webinar, and we are going to switch to the next speaker, whom is Lorenzo Cipioni. So Lorenzo obtained his master's degree in physics at the University of Pisa in Italy in 2014. And then he did a PhD in bioengineering at the University of Genoa, working at the Italian Institute of Technology under the supervision of Professor Alberto Diaspro, whom is famous in the field of optics, and also Professor Luca Lanzano. After that, he joins the Laboratory for Fluorescence Dynamics at the University of California, Irvine in the United States under the mentorship of Professor Enrico Grattan. I guess also most of you know Enrico Grattan, as well as Michel Digman. And Lorenzo now came back to France and is now a team leader at the Centre de Recherche en Cancérologie de Toulouse in France, where he currently applies the advanced microscopy and bioimage analysis methods to the study of triple-negative breast cancers. Lorenzo, thank you for giving us the opportunity to hear you. You can share your screen.
Thank you very much, Cyril. Okay, I think should be good to go. Almost. All right. All right. So hello, everyone. Thanks, Cyril, for the introduction. And so today we're going to talk a little bit about the behind the scene on how the precursor of the dynamic profiler, which is the comprehensive correlation analysis, and how it was developed. So I'd like to start a little bit, not too far away, from an historical kind of perspective, because fluorescence fluctuation methods are not like the standard microscopy methods. So I'd like to give an overview. And you might be familiar with the Brownian motion, which is typically the motion that particles undergo when they undergo free diffusion. And this was discovered, let's say, by botanist Robert Brown in 1827, when he saw under the microscope that pollen grains in water jiggle, basically. Why was that surprising? Because at the time, they had this notion that everything that moved was alive. And then what they did was, as a control experiment, they tested the same pollen grains, but they would be stored a century earlier. And they saw they were undergoing the same motion. So that started a lot of philosophical questions that are not the object of the lecture, of the talk today. But essentially, people started to ask, like, why would that be? Why something that was not alive would still be able to move? And eventually, they figured out there was heat, there was energy that would give that would change the properties of this motion, and as well as particle size. So they started characterizing a little bit more this process until Einstein, you might have heard about, gave this new kind of point of view where it was saying that you have invisible atoms in a liquid that constantly bombard these suspended particles. And therefore, if they are larger, their motion will be slower. If they're smaller, they will get shifted a larger amount. There were, of course, detractors of this theory, saying that particles have no business in the in the thermodynamics world, and that prompted a second paper proposing an experimental way to test whether or not he was right. This experiment was actually conducted by Jean-Baptiste Perrin that confirmed this model and earned him also a Nobel Prize, which is always nice. So following up on these discoveries, also technology followed suit, and they start the application of Brownian motion actually applies to many fields. And what we care about is actually started here, started in the 70s when there was the development of fluorescence correlation spectroscopy that eventually ended up in the first commercial instrument to perform SCS, which was also a Zeiss instrument, leading to the study of molecular dynamics in addition to normal imaging methods. So what information can you extract from fluorescence fluctuations? You can, for instance, have an average number of molecules per illumination volume, therefore concentration, let's say. And although if you look at how many molecules occupy a specific volume at any time, that number would fluctuate because you will have a relatively random number of molecules in your illumination volume. That follows a Poissonian statistics. So, and that, of course, if you want to extract concentration, that will depend not only by the number, but also by the size of the volume. And since it follows the Poissonian statistics, you can directly measure the number of particles that on average occupy the volume by calculating the variance of your fluctuation. Now, this has been actually applied by a very demanding experiment. So in this case, these researchers just stood at the microscope watching a solution of colloidal gold nanoparticles, and they were manually writing down how many of these particles they would observe at any given time to see if they could get something out of it. And now with our tools, we can actually plot this fluctuation, and we can actually analyze them, extract it, like confirming that they follow a Poissonian behavior. And we can also compute the autocorrelation function, which is the mathematical basis of fluorescence correlation spectroscopy, and measure over a century later what was the diffusion coefficient, for instance, of this part. Now, what are the processes that cause a fluctuation? Right now, I talked about Brownian motion, so, but also you can engineer your fluorescent tags to extract multiple other information like conformational dynamics, rotational dynamics, blinking, and other photophysical processes, and many, many things. So what we wanted to do was to try and get as many fluorescent fluctuation techniques in a single analysis. And since from the development of FCS, especially in the past couple decades, there's been a huge a number of new methods based on fluorescence fluctuation, both like spatially, temporally, spatio-temporally, and so on. So we wanted to give researchers a tool to have access to as many techniques as possible to get a more comprehensive view of the dynamic system. In particular, I would say the two extreme technologies are IMSD, which is image-derived mean square displacement. If you're familiar with single particle tracking, you might know what mean square displacement is. And essentially is this series of curves that you can extract, plotting the mean square displacement as a function of the time lag. And that gives you information about whether or not your molecules are undergoing free diffusion, confined diffusion, transient confined diffusion, and so on and so forth. And you can extract quantitative information about diffusion coefficient, the strength of confinement, length of confinement, hopping probability, and so on and so forth. Now, this technique, IMSD extracted the same curve, but has no single particle requirements, like in single particle tracking, so you can use it at the population level. And on the other extreme, there's FCS that analyzes one individual point at very high speed. So you have a very high temporal resolution, but no spatial resolution because you're imaging one point at a time. Now, also the information that is extracted is kind of complementary. So we have, for IMSD, we have possibility to determine diffusion modality, which is very difficult to achieve with FCS, although it happens at a spatial scale that is quite large, because you have to sample this fluctuation across space and time. Whereas FCS doesn't have the same power to exactly pinpoint what kind of dynamics your probes undergo, but it can do it at a special scale of approximately the solution limit. So we have pros and cons. So what we want to do is essentially take the best of what works, being able to get the information about diffusion modality and all the information that comes from FCS at actually below the resolution limit, because we're so the techniques that we implemented in our frameworks are FCS. And FCS works by positioning an illumination volume that created with the laser and observing the fluorescent molecules going in and out of it. Every time a molecule crosses the illumination volume, you get these spikes. And you collect this time trace, you compute the autocorrelation function, which will look in log scale something like this. From this curve, you can extract, for Brownian motion, you can extract the diffusion time, saying, measuring whether or not your molecules are diffusing faster or slower. And you can extract the average number of molecules per observation volume, which is inversely proportional to the height of this curve. We can then get information about diffusion coefficient, concentration, and using a technique called brightness analysis, we can also get the oligomerization state of the probe that is diffused. So it's already powerful as it is, but if, as often happens in the cells, you introduce obstacles to diffusion, then what are structures? What you will measure is lower diffusion coefficient, but you will get no information about what kind of structure is impeding the motion. So another technique which is similar to image mean square displacement is called spot variation FCS. And relies on measuring the same autocorrelation function, the autocorrelation function in the same point, but with multiple size of the point spread function to obtain this kind of. How does it? So imagine that you have an illumination volume that is very, very small, smaller than the size of the structure. Then you would measure essentially free diffusion, right? Because the molecules will have no space to interact with the structure. But if you increase the size, then you would get a bit of free space, a bit of structure. So you would get an apparent diffusion time that would get longer because the probe had before exiting the illumination volume has to go through the maze that is the structure that's blocking motion. And if you take a very large illumination volume, that will essentially, you would see more than free diffusion, you will see the time that it takes for the molecule to cross all these obstacles. And depending on whether the probe is trying to get through impermeable obstacles, dynamic partitions, or meshwork-like environment, you will get different curves. This method is a bit challenging to achieve technologically, because usually, like historically, to get different volumes, you would basically switch objectives. So it can be long and time-consuming. And therefore, it's a similar method is IMSD that essentially has the same concept, but over time. So how does it work? So if you image very, very fast, and you consider what is the jump length, how far are the molecules at time 0 versus time 1, let's say. If you consider very short time points, it would mostly be free diffusion. If you it doesn't change, of course, for free diffusion. So if you have a structure, you consider very short intervals, it will essentially be like free diffusion. If you consider very long intervals, it will give you the diffusion coefficient across the obstacles, and in between, you will have some intermediate behavior. So these are the same curves that you see in square displacement, so you can extract all this kind of information. The last technique that we wanted to implement is called pair correlation function. Pair correlation function was developed by Elisabeth Hinde, that studied the diffusion of GFP and transcription factor across the nuclear membrane, and essentially relies on measuring correlation between two or multiple points. What happens is that if you have if you measure the cross correlation between 2 points that are very close, you will have a high correlation. And but if you place these points farther and farther apart, you will see that you would get a peak essentially at some point in time, which is the time that it takes for the molecules to diffuse from point 1 to point 2. Okay. If you are able, for instance, with imaging to measure the correlation across multiple points, you can actually calculate all of these times and make a map on the obstacles to diffusion. For instance, if you have a barrier between these points on the left and these points on the right, you will measure no correlation between these 2 points, but only between these two. So even though the obstacle is invisible, you have a way to detect it. And the same if something is just slowing down your diffusion, you would get a longer transition time as you would expect if there was no obstacle. And this measure, this is quantified by connectivity, which is whether or not the points are connected, and anisotropy, which is the directionality of the motion. Now, after this long introduction, let's get to the ARI scan. The ARI scan gave us an exciting opportunity because it's kind of I'm missing the word. Anyway, a mix between a point detector and a camera, right? So we can have very fast acquisition in the order of the microsecond. It has a 2D pattern, so we can actually consider it as, in a way, as a micro camera. And it can achieve super resolution by the fact that each of the individual detector is 0.2 LEDs. So it acts like a closed pinhole. And we can actually extract the information about from each single detector. So, for instance, spot variation. How can we achieve spot variation on the ARI scan detector? This is actually quite easy. So if we take the information from all of the detectors and we compute the autocorrelation function, it will be like imaging through a pinhole of 1.2 LEDs, and it will have a certain size. Now, if we disregard the outer circle, it will be below 1 LED, the size of the pinhole. Therefore, it will be a bit more super resolved. And then we can shrink it further and further. And essentially, we have 4 virtual pinholes on which we can that we can use to calculate 4 simultaneous autocorrelation functions that are these, right? But they're relative to different volumes, which is essentially the point of spot variation FCS. So in a single measurement, we can get very easily what was very complicated to achieve with spot variation that was due to changing objective and so on and so forth. And as you can see here, if we start from the black curve, which is the 1.2 LED unit pinhole, and we progressively close and close and close, we get a higher height, a higher autocorrelation function, therefore a smaller number, demonstrating that we're actually shrinking the volume of observation. This is the exact same sample, same data. Now that we have all these 4 volumes that correspond to these values that you see on the left, how do we extract the diffusion modality? What we can do is essentially we do a linear interpolation of these 4 points and see whether it crosses exactly in 0, 0. That would mean that our molecules are undergoing free diffusion. If the intercept is positive, it would hint at a diffusion in microdomains. If it's negative, it would be a diffusion in a meshwork-like environment. We can do the same thing, not with the diffusion time, but with the average number, and see whether the molecules tend to aggregate or be excluded by the obstacles. The more we shrink the volume. And we can use it to construct this kind of graph where each point corresponds to a different diffusion modality. For instance, diffusion in meshwork will have a negative intercept, blah, blah, and then exclusion or aggregation. The farther they are from the 0, 0, the stronger will be the effect of exclusion aggregation or the strength of confinement of the meshwork or microdomain. Image mean square displacement was introduced in a ring-like fashion, correlating at different directions, like on this direction, this direction, this direction, and then averaging them if the diffusion. And that will give us information about confined diffusion, free diffusion, super diffusion, and so on and so forth. And pair correlation was introduced by correlating the central pixel with the first ring, second, so on and so forth. To get basically this kind of functions that Michael showed before. So for isotropic motion, you will have perfectly circular function, and if obstacles to diffusion are present, it will be squished. Now, how was that implemented? In this case, we used groups of 3 pixels of 3 detectors rather than individual detector to increase the signal-to-noise ratio. And you will have one here, for instance, one here, and so on and so forth. Now, to prove that it actually works, we correlated the fluctuation in the black detector with itself, blue, the red, green, magenta, and cyan. And you see that going further and further and further, the diffusion time gets progressively larger because we're measuring correlation at increasing distances. If we consider all the possible connections between these groups of detectors, we can get a connectivity map of how our molecules diffuse within essentially our illumination body. This is an example. So you have, these are the lines connecting different groups of pixels, and this is the anisotropy. You see, this is a sample, is in solution, so you have no difference, everything's flat. But when you go to cells, you will get something like this. You have portions of the detector that will be darker, or like darker blue or redder, showing anisotropy within the illumination volume. And if you look at the anisotropy, these arrows actually show what is the path that the molecules take. So we can appreciate that here we have a slower diffusion, no anisotropy. So that means, so this would be basically an obstacle that slows down molecules. And indeed, the molecules that do not get inside this obstacle flow. That's the meaning of it. And what we used it for was to characterize the diffusion of free GFP in different cellular compartments, what we call unstructured and structured cytoplasm, which is basically the unstructured cytoplasm is where the signal from the GFP is unstructured. So we don't have as many obstacles as you see here, due like organelles, cytoskeleton, and so on, that typically become increasingly prominent around the nucleus. And then we have a nuclear marker to tell us where is euchromatin, where we have a lower signal of the nuclear marker. Heterochromatin, we use, in this case, the NHTT3 cell lines, which is a mouse cell line that has these distinct granules that are heterochromatin foci. And then the nucleoli, which are known to be quite dense. And then we measure all these properties in and put them together. We saw that in all, this is like GFP in solution for reference. And we see that all of these compartments localize in this quadrant. Therefore, we can say that the diffusion in the cell is a microdomain-like diffusion, and obstacles in the cells tend to exclude the proteins, which makes sense. And if you see here, we have these two are the cytosolic compartments, structured, unstructured, and structured. And up here, you have the nuclear compartments. So you have euchromatin, heterochromatin, and then the nucleoli. And you can see that going from the nucleus, the cytoplasm, to the nucleus, we have a consistent increase in exclusion, meaning that there are more regions in the nucleus with respect to the cytoplasm that are inaccessible to the diffusion of GFP. And you see that going toward denser and denser compartments, you have an increase of the strength of confinement of these microdomains. So you have kind of pockets, dynamic pockets within the chromatin that trap the GFP strongly, the more you go towards denser compartments. So we can actually quantify a bit of these properties from diffusion coefficient, this intercept, length of confinement, and all the other parameters that come from the from IMSD and other techniques, so that we can actually monitor, for instance, what is the effect of drugs and other treatments they have on these parameters. So wrapping up, we have a method that can measure in a single point concentration, diffusion coefficient, oligomerization state, environmental organization, connectivity, anisotropy, and diffusion modality from one measurement of 30 seconds. And just a brief 1 minute on what happened from here. During my postdoc at the LFD, I started building microscopes. So now the focus has changed a little bit. So these are a couple of publications that we did there, focusing on highly multiplexing, fluorescence and bioluminescence. And then now we're gonna bring all these baggage of techniques in Toulouse at the CRCT, where we'll apply them to triple negative breast cancer in particular. So thank you for your attention, and it's good to be in France.
Thank you. Thank you, Lorenzo.
Thank you.
So is there any question in the audience? So yeah, I have to remind you that the session is recorded. So if you don't want to appear in the video, I suggest that you ask your question in the chat and I will forward it. Otherwise, just raise the hand and I will ask the question, or you can ask the question if there are any. Okay. So I will start with the first question, except I hear somebody speaking, but no? Okay. Yes. Okay.
Yeah. So I have a question. So this airy disk, is it like a physical pinhole or something like that? I mean, how is a sub-micron resolution achieved with the airy disk? I mean, what kind of system that is? I mean, just to understand.
Yeah. So the in conventional confocal system, you have pinhole first, that you can decide the size, and then you have a lens that focuses the fluorescence on a detector. In this case, the detector itself, it's placed where the pinhole used to be.
Okay. Okay.
And it's engineered to have the size of each detector that is around 0.2 units. And that's kind of the principle of image scanning microscopy, because if you even in conventional systems, you can have a high resolution, a super resolution. Okay? You just have to close the pinhole to fractions of the area. The reason why this is not used much is because the light gets less and less, and the more you do it. So it's not very convenient in conventional systems. In this case, you basically have the same amount of light, but each detector creates a super resolution image. And these images are the only drawback is that these images are slightly spatially shifted one with another. But there are software where you can basically re-overlap them and have a high resolution image with the same number of photons as a 1.20 unit. So high number of photons and super resolution. This is the main advantage of image scanning microscopy.
The particle that you're tracking is still, it's the scattered image of the particle, right? It's not the real particle, because otherwise, is it going beyond the light's diffraction limit or something like that?
Yes, so each autocorrelation function is relative to a volume that is super resolution volume.
Okay, so the image is the scattered image, right? The scattered image of from the particle, right? That's what I mean. And not the actual image of the particle.
Sorry, I didn't get
Answer. The image of the particle that you're tracking is actually the image which is scattered from the particle, right?
No, this
Something
is fluorescence.
It's fluorescence,
okay.
yeah.
Okay.
Yeah, you still need to tag the protein or whatever you're imaging with some. Yeah.
Okay. Thank you.
Thank you. Virginie?
Yes, thank you, Lorenzo. So it's still unclear for me why with these techniques we are not limited by the concentrations of molecules compared to the usual FCS.
Yes, so right. So it's kind of a trick in a way. So imagine that you get your limit for a normal detector is whatever 1 megahertz photons, 1 million photons per second, right? Here, the same amount of photons is actually split over 32 channels, each of which has a limit of 1 megahertz. So you would get more like 30,000, 30 kilohertz per detector if they were evenly distributed. So you could, in principle, go 30 times higher, which would correspond to 30 megahertz for a normal system, but that would be way over the non-reality range.
Yeah, but I thought that also for the FCS you need to see and to measure the fluctuation, and to have fluctuation you should have some molecules going in and out. The volume. And so if you have a very high concentration, there is no in and out anymore. Everything is stable.
Yeah, so that's what. So let's say these are the same guidelines that have been taught, but in my experience, there's not really a limit.
Okay.
That's not really a limit, the concentration. There's actually papers with using very, very high concentration that still not, because it's true that in general, like compared to the average number of molecules, you have less molecules going in and out. But it's also true that you have many, many more photons. So the signal to noise gets better. So, yeah.
Sorry, I just step on that question. I agree, signal to noise gets better because you get more and more photons, but nevertheless, the fluctuation decreases so fast that the precision really gets bad when you reach that level of concentration. Am I right, or?
Yeah, there is like, it's hard. My point is that it's hard to put a number on what is the maximum concentration that you measure.
Okay.
Because it also depends on the diffusion coefficient. So if your model is very fast, you would have more, let's say, events. And in this case, you can still go higher in concentration because you not only have more detectors detecting the same thing, but each of the detectors is so the fluctuation gets more pronounced.
Okay. So we have another question in the chat from Sergei Avilov, and he's asking if you already use the LSM 880 Airy scan for varying spot FCS, and if yes, which software permits you to distinguish between the signals from the individual detectors of the Airy scan? I don't know if this one is for Lorenzo or Michael, but
just a quick answer before Lorenzo, just a quick answer. On the 880, we have no solution in Zeiss company. We have no solution to have a direct analysis for that. We have to extract some results, and after that, maybe we can do something. But I know that there are another software used by the Lorenzo team. It was Airy Scan Global or something like that. I will let Lorenzo answer.
Yes, so the study was done on an 880, and what I was doing was basically saving the result and processing them later. There was no software that was like embedded in the Zen. And basically to do that, I was exporting them in ome.tif, and you will get basically one file per detector. And then you can use the software to get all the to measure the fluctuations, and so on the individual or on the cross correlation, so on. Yeah. And for the spot variation was basically if you see here, the numbering is how it's saved by this file. So the first one would be the central, and then you go sort of like spiraling out. So if you want to do the spot variation, you can just compute the autocorrelation of the central one. And then for getting a larger volume, you sum detectors 1 to 7, and then you do the autocorrelation, and then you add another ring.
Okay. Are there any questions in the audience? Oh. Have you published the software for offline processing of fluctuation from the Airy scan?
This is a so as with Professor Graton, yes, we did publish the globals for Airy scan software. But unfortunately, I've been receiving emails that is not working properly. So I apologize for that. Also because I don't have actually an option, an alternative to do this kind of analysis on actual data. Because it's 7 years ago, plus the computer died. There was not a very there were some technical problems on this, unfortunately. But I'm hoping to have someone work on making a usable version. Let's say it would be probably Python based, but to make a usable version of this kind of analysis, because many people ask for it, and I don't want to let them down. I have no timeline. So.
Yeah. Yeah, starting a team is quite time consuming.
Yeah.
Yes.
I officially started yesterday, so
yeah.
It's
yeah. Okay.
Just an additional comment about that. Just to say that with dynamic profiler, we have access to the raw data for each element of Airy scan. If you want to analyze by yourself the data, you can do it. Of course, dynamic profiler propose to you automatic analysis, automatic fitting, and etc. But you can also have access to raw data.
Okay. Are there any questions in the audience? So, okay, I see nothing. I got two questions for you. One is more asking about a comment than a real question. It's when you address molecular motion, it's always very difficult to make a straightforward and really sure difference between diffusion and flow, and whichever the technique you use, in fact, because it's known that you can have some kind of spurious diffusion that looks like flow, but finally it's diffusion. And so would you comment about the main advantage, and can you give us a kind of a number of how far you can for sure make the difference of what is diffusion and what is flow, in fact?
Right. So with this approach is actually very easy because when for instance, for IMSD, one of the fitting parameters is flow velocity. And that's actually very easy to see because basically it relies on computing the spatiotemporal function, right? The sticks. And that for a system without flow, you would get essentially kind of a Gaussian that would get shorter and flatter over time, but it will stay in the center. It will not move. If you have flow, you will have the exact same
plus motion,
plus a shift toward away from the center. That would give you immediately not only information but also correction for flow, and still a good quantification of the diffusion modality.
Okay. And then can you imagine doing this for different pixels? Maybe I missed some part, but can you imagine doing this for different pixels on the surface of the detector array and do kind of a map of a kind of a tensor flow, for example, and kind of a map of what is flowing in which direction, with which speed, from the upper left to the upper right? And I mean, if you apply IMSD to each pixel within, can you map the diffusion?
So IMSD is a spatiotemporal technique. So you need at least a group of detectors to measure it. But I would, in principle, you could, like in this drawing, you could maybe do IMSD on this group of detectors, on this group of, I would imagine you can. I don't expect huge differences, although it depends on the sample, so I can say.
Okay. And of course, you need to find a pixel position where you can reach the same distance to perform exactly the same analysis.
Right. Yes.
Okay.
Actually, that could be a bit complicated because the size of the observation volumes gets larger the more you shift off the center, because they get a bit aberrated since they're off the optical path. So there's some corrections to do, but in principle, it seems feasible. For flow, it wouldn't matter because it's like flow is shift.
It's more for diffusion,
But for diffusion, there should be each one will have to be corrected with the size.
Okay, then I have a last question about the biological application you've shown at the end was in the nucleus and cytoplasm, and the distances. The size of the meshworks, or somehow meshwork, a restriction to the diffusion you've shown. So it's slide 26, if I remember well.
This one?
Yes, this one. So the confinement area you've shown seems to be somehow always within the same range of value, which is something like, let's say, in between 300 nanometer and 500 nanometer. Is it related to the technique itself, or do you think it's a real biological thing?
So in this context, the length of confinement. So we saw that the diffusion in microdomains. Microdomains is the picture is better here. It's this dynamic partition. So you have these actually diffusing domains that trap this molecule, release the molecules, and so on. So these values should relate to the size. It's a linear size, so you don't know exactly the shape
Yeah. That's an average size. I agree. That's an average size over the volume you are able to observe.
So, in my opinion, it's so these are realistic values if you consider, like, if you look at the nuclear environment, let's say raw materials, right? So you have different levels of compaction of the raw material.
Yeah.
So I wasn't expecting to go much lower than that, because also, like, the size of the GFP is around 4 nanometers, and therefore, like, the most compact areas of the chromatin will exclude altogether, which is actually something that we see. So we see that getting the confinement, but also a little bit of higher exclusion when you go toward more compact regions. So it would be more consistent to something, so kind of a secondary structure of the chromatin domains. It would be interesting to couple it with some kind of super resolution technique to see.
Yeah. That's the ultimate goal of many people. Yeah.
You see, if what we see is what we get.
Yeah. Okay. Thank you very much. So, are there any question left? And if not, if yes, please raise your hand or write something in the chat. And if not, I propose that we stop here. Thanks again, Lorenzo and Michael, for their respective talks today. And I guess this session will be visible again on the website of the GDR, right?
Yeah, so thanks a lot, Lorenzo, Michael, and also Cyril for this nice session. So this video conference was recorded and it will be downloaded on our YouTube GDR Imabio channel soon. And so the next session will be probably in October. So we do a break for summer. So thanks again, and see you soon. Bye. Thank you.
Thanks, everyone.
Bye.
Bye-bye.
Thank you.